We explored early on in this class how theories of sleep changed over time from passive theories to active theories. The brain functions under a completely different set of rules during sleep than it does when we are awake. In this lecture, we will explore these rules. The regulation of sleep, in particular, has been a source of curiosity and study for centuries. Many early scientists believed that gases rising from the stomach during digestion brought on that transition to sleep. Others thought that the lack of activity and reduction in external stimuli during the night was enough to induce sleep. Clues to the actual brain mechanisms responsible for controlling sleep and wakefulness came as a result of an outbreak of a viral brain infection during World War I, encephalitis. A neurologist named Konstantin Vanikonomov, way back in 1917, observed patients stricken with encephalitis lethargica that exhibited some either profound sleepiness or insomnia. He found that the nature of each patient's sleep problem depended on the precise area of the brain that had been damaged by the disease. And he concluded that the brain has different areas for controlling wakefulness and sleep. The study was instrumental in focusing scientists on the brain. This focus, along with the technology to measure sleep, has produced conclusive evidence that sleep is a very active process indeed and controlled by the brain. Interestingly, the brain functions under a completely different set of rules during sleep than it does when we are awake. Some parts become more active, others less active, and some of the interactions between areas change. In a sense, the night shift takes over the brain as the day shift goes off duty, and vice versa. In this lecture, we will explore how the day and night shifts differ. And this begins with a basic understanding of the anatomy and chemistry of the brain. Here's a crash course in chemicals involved in wake and sleep stages. The cells in the brain are called neurons, and you have about 100 billion of them in your brain. When we talk about neural networks, we are talking about a pattern of activation across your neurons. Because the activity of the network of neurons is electrical, we often refer to the network as wiring of the brain. This is a great picture here, the neural network. If you could see the pattern of activity firing neurons across your brain when thinking about an apple versus a banana, you would see that some of the activated neurons would be the same because both are foods and fruits more specifically. And just as an apple and banana have differences, your neural network representations of each would also differ in some ways. This will result in a unique overall pattern for each of the two fruits. When neurons fire, they can cause others near them to fire or not fire through the emission of chemicals. These chemicals are called neurotransmitters, and when they cause nearby neurons to fire, this is called spreading activation. If my words here have now caused you to think about what you already know about neurons and about spreading activation, you are in fact experiencing spreading activation. A pattern began from my words and then spread to also firing across existing knowledge of these topics. Between the approximately 100 billion neurons in your brain are even more glia cells that help to provide structure and insulation to neurons and create an environment that is conducive for spreading activation, the ability for one firing of a neuron to cause others nearby to fire. Depending on the area of the brain, glia cells outnumber neurons by 10 to 50 times. Glia cells in blood also contain chemicals involved in sleep and in wake. The search for one sleep substance regulator, or chemical, you might say, was soon found to be in vain. Just as it became clear with the Human Genome Project that a gay gene was an oversimplification, many would be involved, it became clear over time that a whole cocktail of chemicals interact to produce and regulate the sleep-wake cycle. Kruger and his colleagues in 2009 named them collectively sleep regulatory substances. Many chemicals and their functions are now known, too many in fact for the scope of this class, and some are still being discovered. In the next segment of this lecture, we will explore the major neurotransmitters that are involved.